Well, I can tell you the first part before the slides are up, just what combined brain is. I was going to skip those anyway. I can't see. Can somebody tell me if you guys are working on the slides? You are. Okay, cool. Combined Brain's a consortium of almost 120 different foundations, neurodevelopmental disorders, all working together, sharing resources, data, and trying to help get from diagnosis to cure. And guess who's the chairman of the board? Amber Freed. How about that? Here we go. Okay, now let me see if I can advance. Good, because we can skip the first one, the second one, the third one. We have a lot of very good core values. And now I can get to my talk today, which is how do we streamline clinical trials? You just heard what life is like for a family with a kid with SLC6A1. It's not a simple thing to ask a family like that to travel somewhere to go to a clinical trial. Not once, but repeatedly and repeatedly. Or to do tasks like, oh, just come in and get an EEG, or just do a blood test, or just do this or that. It's really not an easy task. So how can we do what we've learned all about clinical trials? How can we do it better and take care of families better so that when we actually try to study treatments for them and cures for them, we don't burden them and we actually measure what needs to be measured? So basically, what I'm going to tell you about today is I'm going to talk about natural history controls as a really important way to study rare diseases. Then I'm going to talk a little bit about biomarkers. Then I'm going to talk about natural history controls. Then I'm going to talk about surrogate endpoints. Then I'm going to talk about natural history controls. Then I'm going to talk about decentralized trial design. Then I'm going to talk about natural history controls. The point is, we need a lot of natural history to be able to have natural history controls. So I often hear from industry partners that the type of natural history that's collected cannot be used for controls. I've heard that from a lot of industry partners, and I've heard them say that the FDA won't accept the natural history that is being collected as controls. But then when I go to the FDA and talk to them directly and say, look at this, would you accept this as a control? They say, yeah, we would, but industry never presents it. So there's a disconnect there, and we really need to work together between industry partners who are working on clinical trial design and foundations and clinicians who are collecting natural history. So I had the good fortune to be part of this National Academy of Science study to try to make the process of getting drugs approved through the FDA faster, better, and more efficient and probably more accurate for rare diseases. And one of the main, and it came out, it's a book. It's like 350 pages. You can get it for free if you want to read it on the internet. All you have to do is Google National Academy and Rare Diseases. But I'm going to tell you two of the main recommendations that it made that really matter. One of them is the use of alternative and confirmatory data. That used to be called supplementary data. We're changing the name for it because it's not extra. It's data that you can actually use to confirm and approve drugs. And this has been confirmed by a lot of experts, including the FDA. They will accept alternative and confirmatory data in drug approval process. But industry needs to submit that, and we, the community, needs to collect it to be able to do it. The other recommendation it made was to, and I'm really not going to go over these, but I do encourage you to read the 350-page book, is that we need to improve collaboration. We need to bring more patients into every facet of the drug development process, and we need to advance regulatory science. That means try to make these clinical trials better. Try to make them better for everyone and more accurate. So we made those recommendations through the National Academy of Science report, and immediately after that, and maybe it's a coincidence, I don't know, but the FDA released their new effort, which is called the Rare Disease Innovation Hub. And that Rare Disease Innovation Hub is actually an attempt to make the FDA work better for rare diseases and more collaboratively. All of us, the FDA doesn't know what it wants the Rare Disease Innovation Hub to do. They really don't. I've asked them. They want input. We can tell them what we want that Rare Disease Innovation Hub to do. So what do we want them to do? One thing is we want them to start accepting biomarkers for SLC6A1. How do we get biomarkers for SLC6A1? We have to collect blood, pee, poo, EEGs, and data, and CSF. And in fact, if there are any families in here who didn't know, Combined Brain has a team collecting all these lovely things except CSF and EEGs right outside in our little area out there, and you're welcome to donate. And you had a presentation last year that showed that EEG has a strong potential to be a great biomarker for SLC6A1. And I had to go in and out to go to the other meetings today, but I saw a couple of presentations today that showed that EEG has great potential as biomarkers for SLC6A1. We need your EEGs in our EEG bank to show that. You also had a presentation by Anna Falzer today, and she accidentally showed the wrong slides. She showed the slides for SYNGAP1, and so I told her I'd throw up the slides for SLC6A1 so you guys could see them. And this is our pre-competitive biomarker project that we did and we only had 15 SLC6A1 blood samples in that project. Only 15. Now y'all can do better than that. I mean, really. You really can. And you can walk into that place right around the corner where you can give your blood right now if you need to. And what they found when they looked at the proteomics in that plasma, they found that there were quite a few proteins that were increased in SLC6A1 and quite a few that were decreased. And so when they went into detail and looked at these, one of the interesting one, and I'm only going to talk about this one, is IGF-1 on the far right. And what you can see is that IGF-1 was significantly decreased in SLC6A1. That's kind of interesting. This is in plasma, not in brain. And even more interestingly, in fact, by age, IGF-1 goes down even more strikingly over age compared to controls. These are very interesting. These are preliminary analysis. This is preliminary data, but these could be biomarkers. How do we get these biomarkers? You all have to contribute blood, do studies, show up, which you do, I know. So natural history controls. We need in-person natural history. You all already have 3 years of data from the SLC6A1 natural history study. Is that enough? Well, we have more than 18 years of data for Angelman syndrome. I have a 25-year-old son with Angelman syndrome. He's been in natural history studies for 24 years. The first 6 are sort of a separate study. That is not enough. We are still collecting natural history on Angelman syndrome. Is 3 enough? Not even touching it. Natural history studies are the ideal setting to test new measures. If you think that you have a good outcome measure, perfect place to test it is in a natural history study, especially if you share the data or if it's co-owned by the foundation. It's the ideal setting to collect biosamples. So people don't have to walk in all over the country to donate. They can do it at the natural history site. And then it's an ideal setting to be a clinical center of excellence and for future clinical trials. So you saw this presentation, I believe, is on the Simons website. You collect natural history on Simons. Y'all, when you enter your surveys on Simons, there is great data on there that can be shown, but you can't just do it once. You have to do it every single year to be able to look at longitudinal data. Your data can belong to, natural history controls can also be used from Combined Brains Matrix. Combined Brains Matrix has all of these surveys available, and you can fill them out on Combined Brains Matrix because SLC6A1 is a member of Combined Brains Matrix, and I'm happy to tell you more about that later on over a cocktail. Eventually, when you collect enough of these surveys and collect enough of these biomarkers and biosamples, we might be able to develop a surrogate endpoint for SLC6A1. Maybe it'll be a plasma biomarker, maybe it'll be an EEG biomarker, but there are surrogate endpoints that the FDA is accepting. For example, heparin sulfate is a blood biomarker that can be accepted for Hunter syndrome, MPS II. And the FDA is quite open to accepting more surrogate endpoints for rare diseases. And in fact, last year you saw these EEGs for SLC6A1, and what it showed, and I think they were shown, or similar ones were shown again today in Zach Grinspan's talk, that in fact these EEGs were responsive to 4-phenylbutyrate treatment, showing that EEG might be your magic surrogate endpoint. We don't know. We have a lot more to do. So finally, and I only have a couple more things, and I don't know why you're telling me to hurry up, but I still have 3 more minutes. Anyway, so decentralized clinical trials are really, really important. Like you saw with the family who just gave the presentation, what's a decentralized clinical trial? A lot of times I hear from industry, those we can't really do because we can't really trust parents, what a parent says about their kid, or we can't really, it's not really good data and the FDA won't accept it. The FDA tells us they're happy to accept it. What's a decentralized clinical trial? Well, the gold standard is you go into the clinic, the doctor sees you and examines you. Do you think your kid is going to act totally normal that day? Is their behavior going to be on best behavior? They might even be having more seizures. They might actually not do as well on cognitive tests. Do you actually want to capture how they are in the real world? Maybe the in-clinic shouldn't be the gold standard. Maybe the home-based assessment should be the gold standard. And then you can do a visiting nurse or a research coordinator to go to that hospital, even an MD. You can also do home-based EEGs with visiting technicians, and you can do telehealth assessments where you have the physician, looking at the patient and the family right on the telehealth. You can also have patient-entered measures, like there are new EEGs that can be applied by the patient if they have a lot of good instructions on telehealth. You can fill out online surveys, which I know you all love, and you can do that on the Matrix. And then you can also do things like video-based outcomes that, in fact, Combined Brain is going to be working with Casimir that has made a video-based assessment for Angelman syndrome and for Duchenne muscular dystrophy. And we're going to be working with them to increase that to go across as many combined brain disorders as we can. Those kind of video-based assessments are the kind of thing where the mom or the dad or the caregiver takes a video of a kid doing something, like walking across the room. They take it in the same place every time, and then it's scored by PTs or OTs to see whether the kid's getting better or worse. Are those accepted by the FDA? They're already now as secondary endpoints in clinical trials for Angelman and Duchenne. So what are the next steps in all this? Keep collecting data. Everybody needs to collect data and share data. No hoarding of data allowed. No hoarding of data in silos, and everyone, all the families involved. One of the things I hear from people a lot, and working with 120 different neurodevelopmental disorders, I hear people say, I really need you to help me find someone who will cure my kid, but it's really too much trouble for me to fill out these surveys. Okay. Now, come on. If you want your kid cured, you have to fill out the surveys. It's the least you can do. And especially if you want decentralized clinical trials, because then you have to have data on how your surveys have already made sense in a longitudinal setting. Lastly, flexible endpoints. What we found in the National Academy of Sciences study is that you can work with the FDA to look at flexible endpoints. You can try to measure what matters to families. So you might have one thing that matters to Amber's kid, you might have another thing that matters to Kim's kid, another thing that matters to your kid, and they might be different, meaning that they're all in the same clinical trial, but you can't measure exactly the same thing to see if the kid is improved or not. You can consider individualized endpoints, which are like goal attainment scaling. I swear it's the last slide, or almost. And you can base all that on studies that you've made in a conceptual model. Right now, you already have a draft conceptual model, and your final conceptual model is in process, and the data is being coded and will be analyzed very shortly. And that is the end, and I'm only 20 seconds late, and I'm proud of myself. Okay, thank you to everybody.